Today, like I said, we have Karin from EMBL in Germany, and we actually met last year in Crete, which was a really great conference. And also, we kind of connected and realized we're part of the EMBO Fellows Network. So Karin's a successful EMBO Fellow, and we also connected again in October last year. So today, like I said, she's going to be walking through a BioRxiV paper with fibroblasts in the bone. And I think this is actually a really exciting topic, and there's probably quite a few threads that can follow into other areas of research relating to fibroblasts. So with that being said, I think for now, I'll pass over to you, Karin, and we can get cracking.
Great. Yeah, thanks for the introduction. And yeah, indeed, I think it was a really fruitful meeting last year in Crete. And I'm also happy now to introduce a few more people to the Stromal network, because also actually some that joined are from the Zaugg group here at EMBL. So I hope that they also get excited to get a bit more into like the stroma world in general. Yeah, so let me share my screen. So I picked this very recent preprint. It's from a few weeks ago from the lab of mainly of Rebecca Snyder. So she is doing a lot of exciting work in the bone marrow microenvironment, actually quite specifically in the context of bone marrow fibrosis. So they also already had a paper like a few years ago on this in the human context, for example, in proliferative neoplasms. And now they did quite an extensive study in a mouse model. I must say it is a really, really comprehensive work what they did, and I don't show everything. Also, some things might be a little bit too specific to hematopoiesis, and I think here we are a little bit more in a general crowd. Yeah, I think the more, well, partly maybe the technologies, the type of analysis, and also the type of cell types they discovered, I think that would be very interesting to yeah, further dive into. And yeah, I'm very happy if you just interrupt me, ask some questions, share what you think this happens in other organs, right? That would be really exciting. Yeah, and I thought I'd briefly introduce myself in a bit more detail in case you have not met me. So I'm originally from the Netherlands, and I did my bachelor and master's in biomedical sciences and cancer and developmental biology at Utrecht University. And then for my PhD, I went to the University of Zurich in Switzerland with Christian Mosiman, where I did a PhD in developmental biology using zebrafish. So I looked in the most early stages of the mesoderm differentiation, as you can see here in GFP, in this labeled zebrafish embryo. So this contains basically all the progenitors for the stroma we look at. So we discovered a basic gene regulatory program that initiates this first set apart from the rest of the germ layers. And also I looked with lineage tracing in zebrafish into like the early cell fate specifications and traced these lateral mesoderm-derived cells across multiple organ systems. Now, I must say the fish has bones, but no bone marrow. So for this, the zebrafish is not a good model. So then for my postdoc, I decided to go to EMBL Heidelberg and join the group of mainly Judith Zaugg, but also in collaboration with Sinem Saka. And here I started on initiating some projects on bone marrow niche remodeling in the context of myeloid malignancies. So we have multiple projects running in the context of clonal hematopoiesis, myelodysplastic syndromes, and myeloid leukemia, acute myeloid leukemia, and also actually my main collaborator for clonal hematopoiesis and myelodysplasia is also in this call, actually, Boran Gaskes. Yeah, so I'm using a lot of like single cell technologies. We embarked in the spatial transcriptomics field as well. And I'm also do imaging of mode mouse and human bone marrow. Yeah, so briefly, the bone marrow, it's in most of our bones. And as I said, it's not in every species to be found in the bones. It's something that happened during amphibian times. It's a multitasking ecosystem because on the one hand, it supports the whole skeletal system, but also the hematopoietic system. So you can find basically like this interplay between bone homeostasis with this multipotent or mesenchymal stromal cells giving rise to chondrocytes, adipocytes, osteoblasts, and fibroblasts. And then, of course, we have the hematopoietic stem cells in there that form basically all the blood cell types we have in our body. And it's a very active system with renewing basically our cell types the whole day through. And then, of course, we also have a strong immune component in the bone, of course, derived from the hematopoietic system, but also involved in many lineage decisions, for example. Actually, in this paper, they don't touch basically at all on the immune system, so that's nice for us. But for example, in the Zaugg group, we're quite a bit looking into various different immune cell types, actually, especially in the human context. Yeah, so generally in this field of the bone marrow, we are curious, how does this stem cell niche get disrupted during aging and hematological diseases? And it has been always a bit of a difficult system to study because it's inside the bone. It's soft versus very hard. And for humans, it's really hard to actually get the material. It's very precious. And our favorite cell types, the stromal cells, are very rare to be found in, for example, bone marrow aspirates. Mouse is much better, but also there, I think, yeah, we are still on the road to discover a lot of details on this system. Yeah, so then this paper is about fibrosis. So as a brief recap, with fibrosis, you get excessive collagen disposition that alters the normal organ architecture in the niches, and it also influences the normal organ function because of that. And we can find fibrosis in the bone marrow upon hematological malignancies. It depends. For example, acute myeloid leukemia, it can happen, but it's more rare. But for example, with myeloproliferative neoplasms, it's very common. So this is a chronic blood cancer that is often driven by mutations in either or all of these three genes I list here, JAK2, CALR, and MPL. And then generally these patients have overproduction of red blood cells, overproduction of platelets, and a big majority of the patients have this primary myelofibrosis, also called PMF, where they have excessive fibroblast activation. Just like a little clinical note, if you have very fibrotic bone marrow, this like general diagnosis with bone marrow aspirates can then not really happen because it's a really stiff tissue, so they need biopsies, and it's a little bit more complicated. Then a little bit more anatomy on the bone, as this might be not very common for all of you. So we can recognize this then easy schematic that we can easily recognize also in a mouse. So we recognize these ends of the bones, which we call the epiphysis. So this is where the joint is and where the growth plate is. And then we have the metaphysis. This is this trabecular bone. So there are a lot of like small bone pieces that are like interconnected. And this is the area from which the bone has been growing during development, basically. Then we recognize the diaphysis, which is the central part of the bone, the central marrow. This has mainly compact bone and way less of this trabecular bone, and you will later see some images of this also. And also, there's a lot of vasculature running through the bone, and we can always recognize a central artery also. And there are some nice reviews that also have been looking into what are these specialties from these different niches. So there's niches that are here close to the bone. So either here the metaphysis or the diaphysis close to the bone surface. They're seen as generally lower in oxygen because they're further away from the main arteries and arterial structures. And then the central bone is more oxygenated, more vascularized, and also has a more active blood production. Then in this preprint, they're using a few mouse models to label different stromal subsets. So I thought it would be good to introduce them. So they are using CRT2 lineage tracing. So this is an inducible lineage tracing system using tamoxifen or 4-HT. So they have this PTGFRB, which is seen as a more pan-mesenchymal marker, which is also very familiar in other organs. Then they have GLI1, which is mainly perivascular and endostil, so closer to the bone. And then GRAM1, which is an osteolineage. So this is basically what they have. So they have a QWERTY2 line, and they cross that to a ROSA26, which is a lock-stop-lock system that if you treat with tamoxifen, the stop codon gets cleaved out and the cells become, in this case, red. They have the TD tomato. And they followed up the mice at least 3 weeks after tamoxifen treatment. And then here they have an inducible bone marrow fibrosis model. So they do an injection, a transplantation of this thrombopoietin overexpressing hematopoietic stem cells, also called TPO-OE. So TPO is thrombopoietin is an MPL ligand. So it kind of mimics the MPN with the driver mutation in this MPL. Throughout the paper, they here and there threw in another fibrosis model, but for now I skipped that. I thought it became a bit too complex. But yeah, this recapitulates also kind of the human system. And what they need to do is to irradiate the mice first. So there's something to keep in mind. It's quite drastic to the bone, right? It remodels already the bone. And then they would have this lentiviral transfected hematopoietic stem cells injected. Yeah. And then the technology, so they mainly do single RNA-seq, they do imaging, and they do a little bit of spatial transcriptomics at the end. I mean, it's not so much, but it's already tricky to do spatial transcriptomics on the bone. So it's cool that they already managed to do something with it. And just briefly, because single RNA-seq we are all very familiar to, I think now, but Visium maybe is not yet very familiar. So just as a recap, this is a capture slide which contains these barcoded spots here visualized with different colors. And that contains an oligo that has a poly-DT sequence. So this poly-DT can bind to poly-A-tailed mRNA. And in this way, the mRNA is captured from the tissue and will be like covalently bound to the Visium capture slide. And then this can be sequenced. And the resolution will be the resolution of the spots in this case, which is 55 micrometer in diameter. So it's not single cell resolution, it's more areas. So you could say it's like little spots of bulk RNA-seq, basically. So yeah, in the first figure,